currently Principal Research Scientist with the CSIRO, where he conducts research on freshwater and estuarine fish. Since the early 2000s, in positions both with SARDI and the CSIRO, he's been investigating the ecology and population dynamics of fish in the Chowilla region. He's a strong collaborator with different research institutes, management agencies and the community, with application of this knowledge to management of the Icon site and to the Southern Murray-Darling Basin more broadly. Please welcome Brenton Zampatti.
So, yep, I'm talking about the 4 C's in Chowilla. Maybe Chowilla's the 5th C, but cod, cullet, concrete and carp. Themes that Chris has covered as well. And looking at, we keep saying it, 20 years of fish research. It really is amazing, like 2 decades. I saw Mike Harper here tonight. The first time I went to Chowilla, I think, was in early 2004, and I went out there with Mike Harper and we drove across Punker Island, across to Slaney Creek, and I just thought it was an awesome place. It seemed really isolated then, and I must say over the years now it doesn't seem so as isolated. You'd go up there, 2004, 2005 and hardly see anyone all week, and now there's people everywhere, but it's still a beautiful place. All right, so we're talking about management of the site. I've got to talk about the people who've commissioned a lot of this work and who I've worked with for a couple of decades. This is just a few of them, but Tony and Ali, it has been 20 years that we've worked together as a sort of a management science team, and a great sort of close collaboration. And Jan, it's been a fair while as well, I think. And then there's been ecologists like Grace and all the people that came before her. So yeah, Tony's come out in the boat a few times. Ali and Jan, you guys are just running around on the floodplain doing floodplain stuff, I think. I don't know. And then I have to talk about the SA Water Boys because, and they are always boys in the main. Back in 2004 or so, I had a conversation with Daryl Jones and Brenton Erdman when we started working at Chowilla, and they said, don't drive in and out of Renmark every day. Why don't you sort of pitch camp here on the Lock 6 grounds? And that was 20 years ago, and they've never got rid of us. So this is our camp at Lock 6, high and dry during the floods. And it's a great place to stay. Even when I'm not working up here, I sometimes come up for meetings, I go and stay out there. I don't think we're a pest, are we, Warren? If we were, Warren wouldn't do this for us. And like sometimes in the middle of winter, we've come home and the fire pit has been lit and there's hot dinner on the back of Warren's ute. So, yeah, these blokes are legends. All right, a little bit of historical context. And then I'm going to talk about 3 species. I'm going to talk about the Chowilla regulator because I'm an aquatic ecologist and the way it functions that is important. And then a little bit about future considerations. Chris has talked a little bit about this, but I am interested in historical stuff and historical descriptions of the River Murray. And the River Murray Channel was a perennial flowing river. It always flowed. There's reports of it drying to a series of pools, but that's actually when irrigation was quite developed in the late 1800s. So all the records suggest the River Murray always flowed, and it was quite structurally complex. In the vicinity of Lock 6, there were rocky bars and reefs, and Charles Sturt noted rapids that they went down in their boat. So quite a different river to what it is now, which under low flows is a series of lakes. And I really like this brochure from the 1940s, which is like a bit of a propaganda thing from the River Murray Commission, because they talk about the success of locking the river up and really creating 800 kilometres of lakes where there what used to be 800 kilometres of flowing river. And when we turn a river into a lake, the ecological processes change and the animals that live there change. So we go from cod to having carp, We go from having Murray crays to having yabbies, and we go from flowing water mussels to the still water floodplain mussel. But as Chris described, the construction of Lock 6, whilst we lost 800 kilometres of flowing river, we ended up with tens of kilometres of that in Chowilla. So it really reinforces the importance of what we've got left. And we weren't the first to discover this in the last 20 years. Back in the 90s, there was numerous reports. Tony, you would have been involved in this, and other people, I think. Kevin, there's a rather dapper looking Kevin on the cover of that photo there, that report. But both Brian Pearce and Lance Lloyd, who were fish ecologists, identified that these flowing water habitats were important, and Brian, I think, suggested they were important for the recruitment of Murray cod. And I also read in that Chowilla Fisheries investigations that Brian was sampling Murray cod with dynamite in Chowilla. So some of you might know about that, I don't know. But one of the problems was Brian did a lot of work, but we could never find the data behind it. And so I suppose that leads to some of the work that we've been doing. But the ideas that when he put pen to paper were great. Whilst there was knowledge of the importance of these habitats, in the first step decision, the major objectives were about the floodplain and red gums. And it's a pity Murray cod weren't in there. It really is a pity, but we got the fish in later, which is good. But some of the decisions have ultimately been made because of the first step objectives. When we started sampling in Chowilla, it pretty quickly became apparent that it was important for Murray cod. And you could go sample a creek like Slaney Creek, which some of you are probably familiar with, and you could catch 3 fish like that off one snag. And people used to say, oh, Murray cod are a solitary animal, and nah, they're hanging out together often, and big fish. During the Millennium Drought, we were also doing a lot of work in the main channel of the River Murray. And as Chris said, all we were catching really were large Murray cod. There weren't many small fish. So that's a length frequency distribution there. And there's not many fish below 500 millimetres. Most of the fish were a metre plus. Chowilla, on the other hand, in these flowing habitats, had all size ranges of fish. So there was recruitment in Chowilla throughout the Millennium Drought when there wasn't in the main channel. We saw similar patterns in the Lindsay Mullaroo, and we're at the Chowilla Icon site tonight, and no one's really mentioned the sort of Lindsay system, but it's part of the Icon site. And Lindsay Mullaroo, because it's got flowing water, we saw these similar patterns. So we were interested in what was going on in Chowilla that was different potentially to the main channel of the Murray River. And first of all, we wanted to find out, well, were Murray cod spawning? And this is a little baby Murray cod, a couple of weeks old. And if you look at it, it almost looks like a big Murray cod, but just in miniature. Murray cod lay their eggs in a nest, and when the eggs hatch, the little Murray cod sit there for a while, absorb the yolk sac, and then they move up into the water column and they drift. So you can set drift nets to catch these Murray cod, but they're also attracted to light. And so we would set light traps and drift nets throughout Chowilla to try and work out where these fish were spawning. And we were also doing that in the main channel of the River Murray. And what we found through quite a few years in Chowilla was that Murray cod were spawning every year at the same time, no matter what the flow was. And that's their life history. So between sort of mid-October to late November, Murray cod spawn. And they also were doing it in the main channel of the River Murray. Those big fish were spawning. But it was something about the flowing water habitats in Chowilla that were promoting the survival of those small fish. And once they could get through to a certain size, then they were carrying through to reproductively mature fish. So we knew fish were spawning in certain places in Chowilla. We were also interested in how fish move between certain habitats. So we spent many years implanting radio transmitters into fish, both big and small, well, down to about 500 millimetres, and following these fish around. Many of you would have been out to the Icon site, would have seen that we had these remote telemetry towers. So these tags are setting off a radio signal that we can track manually, or we can track them with these towers. We're also looking at callop or golden perch movement at the same time. And what we found within Chowilla was that there's core habitats within the system that there's high abundances of Murray cod, and Murray cod tend to move between these habitats. Now, you're not allowed to take this information away from this room tonight, otherwise everyone will be going fishing in these places. But really the whole length of Slaney Creek, parts of Pipeclay Creek, certain places within Chowilla, and for those of you who know the system, you'll probably know where those places are, because they've all got large wood and they've got flowing water. And these are these key habitats for these fish. And when some Murray cod don't move much at all. They sit up in Slaney Creek day in, day out, year in, year out. But some fish move between Slaney and Chowilla, some fish move between Slaney and the River Murray, and some of them do it seasonally. So as we were gathering this data, we were putting together a conceptual model of how Chowilla worked for Murray cod. And so we found that Slaney Creek was a key spawning site for Murray cod, high abundances of large reproductively mature fish. When those larvae drifted, they drifted out of Slaney Creek, and they would drift with the flowing water down Chowilla Creek and settle out in the edge habitats. And that would be their sort of nursery habitats. And it's also where their food was settling out as well. Chris showed some data on the abundances of Murray cod over time and showed that there was decreased abundances post-flooding, the big flood years. And what we found whilst we were doing these movement studies is that we said most fish weren't moving a lot during low flow years. And then during flooding, we had some fish moving hundreds of kilometres. Most of the time it was upstream, some of those fish up to the Lindsay Mullaroo, some as far upstream as Mildura. But at the time, each of these radio tags had a mortality signal in them. So if the fish stopped moving or died, you got a different signal and we could find those fish. And about 25% of our tagged fish died. So although we didn't have really large sample sizes, we could tell that, if a big Murray cod in the river during these events, about a quarter of the fish could be dying. And that's why you see this decrease in abundance at the site. Also because fish are emigrating from the site. And what we've found over time is that sometimes fish move out during floods and then they end up coming back a few years later. So there's a bit of a balance up in the population again. All right, our next fish, which I could talk all night about again, but I'll just touch on really quickly, is golden perch, or callop, we call them here in South Australia. Really, they are quite abundant through Chowilla and they are a popular angling species. We get them across all the habitats, but they are probably most abundant in those fast-flowing creeks. Chris showed that post-flooding in 2011, we saw really high abundances of golden perch, and it was driven in large part by young of year fish that were spawned in the year of the flood, but also a whole lot of age 1+ fish that were spawned the year prior to the flood in what was still a fairly low flow year at the end of the millennium drought. And we hadn't seen those fish in the population. So some of you have seen me talk about this before, but we can actually use the ear stone from the fish, the otolith, which we use to age the fish, to also look at where that fish has been throughout its life. Because the chemistry in the otolith reflects the chemistry in the water that the fish has been swimming through. So if a fish has been to different places in the River Murray with different water chemistry, that's reflected in the otolith. So we take our otolith, we cut a section through it, and we get a laser and we blast from the core of the otolith, which is that's where the fish was born, across its life to where it died. That's at the edge of the otolith. We can match that with water chemistry across the basin, and strontium is something we use because strontium in water reflects catchment geology, so different catchments in the basin have different strontium. And for this fish, which was a 12-year-old golden perch, we can track where that fish has been. That fish was born in the mid-River Murray, somewhere upstream of Mildura. Very early in its life, it probably drifted into the lower Murray, maybe somewhere down around Chowilla, spent its adolescence there, and when it reached reproductive maturity, migrated back upstream. So the fish's life is recorded in its otolith. And what we know now for somewhere like Chowilla is that the fish that comprise the golden perch population could be born in the lower River Murray, they could be born in the mid-River Murray, or they could be born in the Darling River. So places a long way away from Chowilla are contributing to the population and this connectivity. Golden perch also move as adults and they move upstream, and we've done a considerable amount of work tracking Callip and looking at their movement out of Chowilla and also within Chowilla. And in any given year, no matter what the flow's like, about a third of the radio tag fish would move out of Chowilla and upstream. But the really big movements are during flood years. And Fred Reynolds back in the 1980s recorded these large movements out of the lower Murray, thousands of kilometres upstream. We've seen some of our fish do that as well during the floods. They've moved from Chowilla to Mungandai up on the Queensland-New South Wales border, which is a long way. So different scales, different life history scales to Murray cod, and something like Chris said, we can't really manage at the icon site scale, but we can do things to help these fish. I'll get to the concrete part of the talk now. The old concrete structures were replaced, fish couldn't get past them. We put fishways on them, so these Murray cod that are moving out of the system when they want to, or these Callip that want to move upstream can now move past those structures. The big concrete structure was the regulator, which essentially is a stop log weir, like the weirs on the Murray. As an aquatic ecologist, this concept was really quite weird to us when it was proposed, because I studied at Adelaide Uni, like Chris did, like some people here did. We studied with Keith Walker, as Chris said, was a doyen of regulated river ecology, and along with a guy called Jeff Petts, they wrote many papers that weirs are bad for riverine ecology. And here we were trying to use a weir to restore ecosystem health, and primarily for floodplain trees. But the concept was indeed, counter to the thinking at the time and highly experimental. And there was quite a bit of commentary on the concept from ecologists, and we had a number of risk assessments undertaken for fish and other animals. And based on these risk assessments, the regulator has been operated in a manner, as Jan described, to try and minimise risks. So whilst the regulator has been operated through our intervention monitoring, we've had some opportunities to test some of these risks and how to mitigate risks. We've been able to look at the drift of Murray cod and of zooplankton, and this study, and I've been showing a few scientific papers here because there's actually been a whole stack of scientific papers that have come out of our research. They've informed management at Chowilla, but they're also being used across the Murray-Darling Basin and globally. So what we're doing at Chowilla has broader applicability. In this paper, we looked at how the regulator impacted the drift of Murray cod larvae and zooplankton. There was also a water quality component, but I'll just look at the Murray cod component. And what we saw was we had this original model of larvae drifting down the creek. When you put the regulator in and you slow water velocities, you try and maintain water velocities, but you slow them a bit, these larvae potentially settle out sooner, and they may not have access to these nursery habitats down the creek. Now, does that impact the recruitment of Murray cod? We can't see that yet. We've collected data, we haven't seen it. Maybe it doesn't, but the mechanism is there that they're not reaching the nursery habitats they used to. So to our fourth C, which is carp. Carp love regulators and they love floods. And we all know how they went off during the flood, but also when the regulator was operated, these huge abundances of carp on the floodplain. We've been doing some work with our colleagues in Victoria, looking at modelling carp populations in the lower River Murray and how they respond to floods and regulator operations. And this plot here shows, with the black line, the carp population in the lower River Murray modelled during times of just no regulator operation, natural floods, and then various scenarios where you may operate Catarapco and Chowilla every year, or you might just operate one. And what this does is it provides us with a tool to try and predict how you may minimise the impact on carp populations of operating the regulators. I think we're at a stage now after 10 years and 6 regulator operations where we can hopefully start to put a lot of the science on the table and really consolidate this to inform future management, not only of the Chowilla regulator, but regulators that are proposed elsewhere. I think at the site we could really be using Murray cod and more broadly as a flagship species. You know, we've shown tonight that these fish operate over scales of tens of kilometres, the importance of flowing water. Somewhere like Chowilla would provide an excellent model for a freshwater protected area. We don't really see many globally. We have marine protected areas, we have terrestrial, but you know, we could put this place up in lights and say we need to conserve these habitats for Murray cod. Couple more opportunities. I think the concept of using traditional ecological knowledge and Western science, it's often talked about these days. I think we should be exploring it. Somewhere like Chowilla, it would be really good to be looking to that into the future. The greater integration of community and science. In the early days of working up here, I think as fish ecologists, we had more interactions with the community reference group. I'd like to really sort of reinstate that and have some more of that. But importantly, we need to look bigger than just the icon site. Well, we've shown that golden perch operate larger, but many riverine processes operate at scales larger than the icon site. And over the past couple of months, I've spoken at a couple of icon site sort of celebratory events, and it's got me thinking more and more about how this isn't the River Murray, this is the Colorado River, but we manage patches in the landscape. Icon sites are patches in the landscape and we can put water on them and they go green. But how are we interconnecting these patches in the landscape? And how are we using rivers to connect them? And I think we really need to think more holistically about how we're managing our icon sites and the river system as a whole. Some of these other icon sites as well have got projects, regulators that are being built that may mean less environmental water in the river because the regulator is being, or pumping is being used to do more with environmental water, and more water may be left in the river for consumptive use. So what does that mean for end of system flows? Does the Klim icon site miss out because the other icon sites are having supply measures integrated? And again, I think we really need to sort of just raise our vision a bit. The icon sites are a great concept, but how do we integrate them? How do we manage the riverine ecosystem as a whole, really, and for end of system outcomes to get water down the end? So that's it from me. Thank you.